Hello everyone, welcome to the course on computer-aided drug design. We will continue on the topic of target-based drug design. That means target could be a protein or an enzyme, and you want to look at how the ligand goes and binds to that. Okay, so in the target-based approach, we need to understand this particular point. Okay, if you look at the protein data bank, it has 3D structures of 50,000 protein structures, which is 1% of 5.5 million protein sequences. That means two-dimensional sequences available in this SwissProt, and which is 0.00007% of Earth proteins. Okay, because Earth has a lot of organisms, genes, so we have very little knowledge about the sequence of proteins found in the Earth, and we have still very little details about the three-dimensional structures of the protein. So there is a lot of scope for the next several hundred years. We may be looking at 2D sequences and 3D crystal structures and so on, if you want to look at proteins involved in the disease. Okay, so that's still a long way off. Okay, let's look at this particular database called SwissProt. Okay, SwissProt. Okay, if you type, this is called UniProt. Okay, UniProt, there are a specific ID for each one of the protein. We can get the two-dimensional sequence of all these protein. Let's for example look at this. This is a lipoxygenase, human lipoxygenase. Okay, as we have been telling, this 5 human lipoxygenase is involved in the arachidonic pathway of the inflammation, and it leads to bronchitis and so on actually. So let's look at the sequence of this, and we will make a comparison of other sequences of other protein that are found, okay, in the UniProt. Okay, so we give the UniProt number, and then we say run BLAST. Okay, BLAST is a basic local algorithm search tool, and it looks at other proteins found. Okay, job is running here, so it will look at two-dimensional sequence of other proteins which are similar to the sequence of this particular phylox human which I had given. So it will take a long time. I had captured the screens before for quick. Okay, so this is how we will get. Okay, this is the protein, this is the 5-lipoxygenase. It's called arachidonic 5-lipoxygenase because arachidonic acid is the substrate of this, and this particular protein is from Homo sapiens. So it compares other proteins in the UniProt two-dimensional sequences and says this particular protein, okay, from Papio anubis has a sequence similarity of 98.2% identity, and then another one, gorilla, 98.5, and then another one 98.5, and so on. So as you can see, it makes a comparison. It gives something called e-value, score, identity, okay, and so on actually. So we can look at large number of proteins, and we can select the one which has a very high percentage identity or very high score. Okay, so we can also look at for example look at this particular one, okay, 98.2, and they make a comparison of the 2D sequence alignment. Okay, so you can look at here. Okay, it's got an identity of 98.5, and so on actually. Okay, so let me again check whether the software has done its job. Okay, it's still running, so it takes a little bit long time, and you will get the results here with the comparison of many things. Okay, we'll do that later because it is still running here. Okay, so that's very important for you to identify, is there any protein or proteins which have similar sequence identity as the protein of your selection. Okay, so we have the target sequence, template sequence, then we do multiple sequence alignment, and then if the three-dimensional structure of that protein say like a gorilla or some other animal is available, okay, then we prepare the three-dimensional structure of our protein of interest using that particular template, okay, and then we see whether the protein which we have created is okay using a various bioinformatics checking. Okay, this is called the homology modeling. Okay, once we have that, okay, so we use the software. There is a software called Swiss model. We look at it. I just showed you the BLAST also. So we can do a homology modeling using the Swiss model. Okay, then we can predict the active site of the protein, and then if you have a library of ligands, you start docking all the ligands into that. It's called AutoDock, Gold, Glide, so many different softwares, commercial, non-commercial softwares are there, so we can make use of them, and then we get something called a docking score, which tells you how good the interaction is. We can also do molecular dynamics. So that is how the target-based drug design happens actually. Okay. Okay, so as I showed you here, what do you do? So we have a sequence A, say imagine 322 amino acids, sequence B 450 amino acids. So I just showed you BLAST. So it makes a pairwise alignment of both A and B. 100 amino acids are identical, so it is 100 by 320 because 320. Generally we always use the smaller sequence length as the denominator, so identity will be 31%. Also 23 amino acids are different by conservation substitution, meaning that the chemical properties are maintained. Okay, so we add 100 plus 23 divided by 320, that comes to 38.4. So that is called similarity. Identity is exactly matching. Similarities, we also add other amino acids which may have similar chemical properties. So sequence identity is 31%, sequence similarity is 38.4%. That is how the softwares like BLAST calculate. Actually, I took this calculations from this particular reference. Okay, so in target-based drug design, we identify target identification validation. That means if I know the disease mechanism, I decide on which target to focus on. We are talking about inflammation, arachidonic acid pathway. So your target could be a lipoxygenase, which produces leukotrienes, or it could be prostaglandins like COX-2, or prostaglandin E synthase towards prostaglandin. And then we should also, of course, have experimental approach, assays for measuring the interactions between both. And then we need to do the virtual screening. Take database of ligands like all your zinc or drugs, data bank or so on, and then keep docking them, and then find out how docking happens. We can also do a QSIR, refine the compounds, and then we can use Lipinski's rule or other rules, shortlist compounds, then go to your animal studies, in vitro studies, clinical trials, FDA approval, and so on. So this is how things happen. Now, if you look at proteins, their interiors are tightly packed. Okay, if you take water, 36%, cyclohexane 44%, ethanol 47%, closed packed sphere 71%, protein interior is 75%. That means proteins are very tightly packed interiorly, so the ligands do not actually penetrate into and bind. Generally they go up to some active sites, mostly on the surface, okay, or on the leaf or gap between two protein and so on actually. So if you look at van der Waal interaction of a CH2 group with three environments, okay, so CH2 interacting with water, you get minus 1.8, CH2 interacting with cyclohexane minus 1.9, CH2 interacting with protein interior minus 3.1. So interior is generally very hydrophobic because protein is generally taken in aqueous medium, so hydrophobic regions are always buried inside the protein. Okay, so that is another point you need to keep in mind. So how does hydrogen bonds improve the stability of protein? If you look at serine, alanine, this is about minus 0.9 kilocals. Okay, so valine and threonine minus 1, tyrosine, phenylalanine minus 1.2, and so on. Okay. So this is how the interactions between the hydrogen bonds in various amino acids contribute to the energy. Okay. So hydrophobic interactions make the major contribution to protein stability. Like I said, the interiors are tightly packed and all the hydrophobic regions are inside. The hydrophilic, that is the regions which interact with water, are outside. Okay, so hydrophobic regions. Hydrogen bonds by side chains and peptide groups leads to protein stability, of course. Okay, disulfide bonds also are there, contribute to protein stability mainly by reducing the conformational entropy of the denatured state. Okay, so it prevents the changes in the conformation of the protein. Okay, so that's disulfide bond. Okay, so if you look at the delta S minus 2.1 minus 3 by 2 R ln N. Okay, so this is the number of conformations that are possible. N is the number of residues in the loop formed by the cross-link, and N is the number of residues in the loop formed by the cross-link. R is the Gauss constant. So the equation predicts the stability will be increased by 3, 4 and 5 kilocals by loops 15, 45 and 135 residues respectively. Okay, so if you have a lot of residues in the loops, the stability of the protein gets increased based on this particular equation. Here N is the number of residues in the loop. Okay, so charge interactions. The charges on the surface of the protein are generally arranged so that there are more attractive than repulsive interactions near neutral pH. Okay, so instead of repelling each other, they'll be attracting each other. Okay, at neutral pH. So they contribute favorably to protein stability. Salt bridges. Okay, so these are oppositely charged groups in protein within 5 angstrom. They are also called ion pairs or salt bridges. So salt bridges also contribute to the stability of protein. Okay, gain in configurational entropy of water on protein folding. That's another point. When a protein folds, much of the water hydrogen bond to the protein will be released. So this gain in the configurational entropy of the water may contribute to protein stability. Otherwise, too much water is there, protein can take too much of conformation. Okay, then 65% of more charged groups are buried in protein containing 700 amino acids than in proteins containing 100 amino acids. Okay, so removing buried charges might be a way of increasing protein stability. So hydrophobic interactions contribute to 60% and hydrogen bonds contribute to 40% to protein stability. So these points are very important because when you are designing drugs, we need to understand that hydrophobicity and hydrogen bond contributes quite a lot than many other types of forces. Forces contributing to instability of protein. The major one is the conformational entropy. That means rotation around many bonds in a protein is much freer in the denatured state than in the native state and provides a strong entropic driving force for protein unfolding. Okay, so if it has got too many rotatable bonds, so once it gets denatured, the rotatable bond leads to a lot of conformational changes, so protein becomes more unstable. So there are several rules when we talk about target based design. Okay, so this is taken from this particular reference. Coordinate to key anchoring site. Okay, so you should identify what are the key anchoring site. Like sometimes they have catalytic triad, for example, in biotransformation. So those are the key. Make use of hydrophobic interactions because, as I said, there are a lot of hydrophobic interactions in protein inside. So we need to make use of hydrogen bonding capabilities. Okay, this is the second. First is hydrophobic, hydrogen bonding, then make use of electrostatic interaction. Favor bioactive form and avoid energy strain. You should try to avoid conformations of the ligand which will lead to higher energies, which is energy strain. So you should look at the drug or the ligand which has the lowest energy strain or in the form of best bioactive form. Optimize van der Waals contacts and avoid bumps. Okay, structural water molecules and solvation. You should look at structural water molecules and solvation. We'll talk a little bit more and then finally consider entropic effect. We'll talk a little bit more on each one of these headings. Okay, so what is this proper anchorage of the ligand? Okay, so we should know the catalytic side based on experimental or theory. So we should adjust our ligand so that they make use of those catalytic side. This positions the ligand in the active site and also counteracts the effect of desolvating the two components when binding occurs. Place a hydrophobic surface of the ligand in hydrophobic sites of the target. Okay, that's very important. Otherwise, there could be a non-polar surface exposed to water. You want to avoid that. Free energy contribution due to hydrophobic forces is 2.9 kilojoule per mole per methylene group and 8.4 kilojoule per mole for a benzene ring. Okay, unlike hydrogen bonds, the hydrophobic interactions are not directional. Okay, like I talked about the pi-pi interactions and so on. Hydrogen bonds are a little bit more directional. Generally, non-bonded interactions are not so directional, but hydrogen bonds are a little bit directional, and hydrophobic interactions not directional. Okay, then unsatisfied hydrogen bond donors and acceptors are rarely seen in proteins and protein-ligand complexes. Okay, because this would be highly energetically unfavorable. So, if there is a ligand which has a hydrogen bond donor or a hydrogen bond acceptor group, you need to position that so that it has interaction with the active site corresponding acceptor and donor and the amino acids in the active site. Okay, so you will rarely have an unsatisfied condition. A carbonyl oxygen is optimally satisfied when it accepts two different hydrogen bonds, okay, close to 120 degrees. This point you need to keep in mind. So if you have a carbonyl in your ligand and if there are two hydrogen bond donors, so ideally if it goes and fits there, then that conformation is very suitable. Hydrogen bonds to carbonyl oxygen atoms with the C double bond OH angle close to 180 form the basis for beta sheet formation, and that is also quite favorable. Okay, the average NHO angle is about 155, 90% lying between 140 and 180. So ideally the NHO angle should be 155. Okay, so if you are designing ligand and if there are hydrogen bond acceptors in the amino acids in the active site, then look for that sort of angle. The protein groups are capable of forming hydrogen bonds like this, where groups are not explicitly hydrogen bonded, they are probably solvated. So you need to remember that if there are some groups which are not hydrogen bond forming, then there could be water coming in and they may be interacting with the water. Ligand protein electrostatics can be achieved by placing a positive charge in close vicinity to an enzyme negative charge. If you are designing ligands, you always find that the ligand takes a bioactive conformation while binding to the active site inside the protein. Okay. Van der Waals interactions occur over a short distance and attraction decreases as 1 r raised to the power 6. Okay, so you need to keep that point in mind. Van der Waals interactions over a short distance, they are not long distance, so you need to keep that point also in mind actually. Attractive van der Waals interaction occurs as the shape of the protein binding site and the shape of the ligand match well. So that means shape comes into the picture, shape of the binding site and the shape of the ligand, they should match nicely. Inhibitor should target the displacement of water molecules tightly bound to the protein. So incorporating elements of water molecule within the inhibitor is good. Okay, so that means the inhibitor should have the capability of displacing water molecules that are tightly bound. Okay, so ideally you should have some groups which can displace that water. When polar charge groups are considered in the design of a ligand, one should leave some room for other water molecules to solvate the charge center, except possibly when a solubility is formed. Okay, a flexible molecule has a better chance of finding an optimal fit into a receptor. Okay, but then if it is too much flexible, it's going to be at the cost of large conformational entropy. So sufficient conformational rigidity is essential. So you need to balance this to ensure that the loss of entropy upon ligand binding is acceptable. A rigid molecule has little conformational entropy, so but it is unlikely to fit optimally into the receptor. So you see that if it is too much rigid, it might not fit optimally, but if it is too much flexible, yes, it can take different conformations, but it will have too much of conformational entropy. Okay. Analysis of contribution of various functional groups to protein ligand binding demonstrate that each freely rotating bond in ligand reduces the binding energy by 2.9 kilojoule per mole. Okay, so if you have 1, 2, 3, 4 flexible bonds, it keeps on reducing by 2.9 kilojoule per mole. So making a flexible molecule more rigid will lead to enhanced activity if the right conformation is maintained. So like I said, it's good to make it rigid because you can reduce the conformational entropy, increase the binding energy, but then the conformation should match with the biological active conformation as well as optimally binding to the active site of the protein. So you need to balance all these if you want to achieve a good ligand structure and shape. So there are 4 basic methods in target-based drug design: analog design, database searching, computerized de novo design, manual design. So there are 4 different approaches by which we can do this. We will look at each one of them a little bit in more detail. So what is this analog design? Chemically related molecules are synthesized and tested to develop 3D structure activity relationship. That's what is called analog. So you make analogs of a parent compound, and then we develop a 3D structure activity relationship based on their structural features and activity, like we said. So a loss in potency is interpreted as a loss of good complementarity between the ligand and the receptor. Okay, so if the potency is going down because of you removed a hydrogen bond donor, then you know that that complementing feature that's available in the active site is not made use of. Okay, that's called the database search. So docking of synthesized or not naturally occurring compounds in various databases and rank their docking score. So this is what we generally follow. Okay, so we select large number of molecules based on, say, Lipinski's rule or other rules, and then start docking them and then rank them based on their docking scores. So it does not require any synthesis as of the previous analog design. Highly dependent on the content of the database. So you have to watch out. If you select one database, maybe it has got only one set of molecules, so you may miss out other set of molecules. Due to conformational complexity of molecules, only few of their conformations are actually stored in the data bank. That's also important. So a molecule can take several conformations, but only few conformations may be stored. So only those conformations will be docked to the active site. So you may miss out some other conformations of the same molecule. That's another. There are thousands of chemical suppliers offering greater than 3 million of organic molecules. So they offer a great diversity and can be purchased and tested rapidly. So database search is a good idea to follow. We can purchase them or even if you can synthesize them later on if they are found suitable in our in silico, and then we can test it rapidly. The goal is not to find compounds with ultra potent activity, but discover subsequently, because they could be the starting for further synthetic modification. That could be a sort of a lead, and then we could modify the structure so that we can improve their activity. Third approach is called the de novo design. Okay, here discover a new chemical framework that fits to the active site of the target receptor or enzyme. Then basically you start on an existing moiety and we keep adding fragments step by step, so that maybe when you add a particular group, it increases the electrostatic interaction, or when you add another particular group, maybe it improves the hydrophobic interaction, or it creates a good hydrogen bond donor capacity. So that's called de novo design. So you start with an existing moiety and keep on improving so that you get a better interaction between the new designed ligand and the active site. Other methods consisting of assembling novel molecules from pieces that are positioned optimally in favorable regions of the active site. So you take 3-4 pieces and place them in different places inside the active site so that they create a very good interaction, and then you create by linking those pieces together. That's called de novo design. So you may come up with new structures, and of course those structures, whether I can synthesize them or not, that's a different story. We may have to check it out later with the help of medicinal chemist or synthetic organic chemist. Manual design. So start from an existing solution which consists of the visualization of a complex. Okay, so you look at the existing solution and then you keep modifying. This procedure is currently undertaken either by the medicinal chemist or the molecular modeler. So we have different approaches. Of course, you can combine some of them together so that ultimately you end up with the most favorable ligand, which has best bioactive conformation, which binds very optimally to the active site of the target. Okay, so there are many drugs derived from target-based approach. Okay, so captopril, this is ACE. Okay, angiotensin converting enzyme. It is involved in hypertension, Bristol Myers Squibb, then dorzolamide, carbonic anhydrase, glaucoma, Merck, okay, HIV protease, nelfinavir, Pfizer and Lilly, oseltamivir, influenza, imatinib, chronic myelogenous leukemia, Novartis. So these are just some examples. There could be many more, but I am saying that it's possible to look at target-based approaches. Okay, so when a ligand is bound to the active site of a protein, okay, you get the binding energy or interaction energy, non-bonded interaction. Okay, so softwares make use of something called a scoring function. They calculate something called a scoring function, okay, based on the hydrogen bond, electrostatics, van der Waals, hydrophobic, pi interaction, and so on, and then they report the scoring function. So higher is the scoring function, higher is the binding or interaction. That's what it is called actually. This is how it is performed. Okay, the force field based, as you know, we talked quite a lot about force field, charm, amber, different MMFF and all that. So we can based on these force field the results, we can calculate a scoring function. Empirical scoring function count the number of interactions and assign a score based on the number of occurrences. For example, number of hydrogen bonds, number of ionic interactions, number of hydrophobic bonds, number of rotative bonds which got frozen because of binding. So then you calculate a scoring function. Knowledge base. Observe known protein ligand structures and favor interactions and geometries that are seen often. Okay, and then if you see your ligand also binds in that form, give a score. Okay, directly linked to free energy because real life distribution, but based on small number of entries. So there are different scoring functions available in the market. Each software has its own scoring function. So the results from one scoring function and another scoring function need not match at all. Okay. So we will continue more on this topic of target-based drug design in the next class as well. Thank you very much for your time.